All right, hello. Thank you for having me chair this session. It is my pleasure to introduce Christopher Shingledecker. He is an assistant professor of physics and astronomy at Benedictine College in Atchison, Kansas. I just found out that he had an astronomy class where my cousin was in. If you meet anyone with the last name Camarca in the United States, we're probably related. After receiving his Ph.D. from University of Virginia, he was an Alexander von Humboldt Foundation postdoctoral research fellow in Germany, where he worked at both the Max Planck Institute for Extraterrestrial Physics in Munich and the University of Stuttgart. His research in theoretical computational astrochemistry with a focus on molecule formation on interstellar dust and ice. He is one of the recipients of the 2017 Rao Prize, and Professor Shingledecker is a member of the Society of Catholic Scientists. The title of his talk is Forming the Molecular Building Blocks of Life on Icy Cosmic Dust.
All right, well, thank you very much for the introduction, and thank you also to the organizers for this beautiful conference, for the opportunity to be speaking to you today. Now, I can't promise that my talk will be as entertaining or as insightful or even as beautiful as the previous talks. In fact, I'll be referencing them a lot. I'm in the cleanup spot here. My slides will also not be very beautiful. They'll be crude and childish, as will my reasoning be, okay? Okay. So, forming the molecular building blocks of life. Okay, I should probably back up, maybe? Okay, I'll back up. Can you all hear me still? Okay, good. All right. Biological molecules are very complex, a little bit like this watch. Though you can probably get them for less money than you can get this watch if you're called Sigma Aldrich. So they're complicated. And I'm an astronomer, so I like to simplify things. Order of magnitude pi is 1, all this fun stuff. Okay. So I'm going to be talking about the building blocks. So the building blocks, much simpler. The material conditions for, not a watch, but life. Okay. So let's talk about origins of life research and kind of get some perspective on some of the questions that we've been discussing and that origins of life research deals with. So one will be, how do the material conditions of, well, not the watch, but life come to be? How do you get the material conditions for that? And the second question that's been talked about, both in the talks as well as some of the questions and comments, is how, under what conditions do you go from the material conditions of life to something that has the form of life, form of a watch? How do you do that? And there's a related question, I think, which is, are the material conditions themselves necessary and sufficient, to be mildly philosophical, for the formation of life? When you have all the material conditions in the right order, do you then necessarily get life or watches? I'm going to cease with the watch analogy at this point because it's too complex. Okay. All right. Keeping in mind that the effect can't be greater than the cause is something. Okay, well, now that I've teased these questions, I'm going to ignore the last two and focus entirely on the first one. So I'm sorry for the disappointment. This will be maybe a less interesting talk than you're expecting. Now, at this point in my talk, I have to acknowledge that proteins and DNA and RNA exist, and I will tell you a little bit about it, and if you're a biologist or molecular biologist or biochemist, you'll probably find this to be, again, a crude and childish overview, but bear with me here. So this is a protein. Does not look like a steak. This is myoglobin, which was the first protein to have its structure determined by X-ray crystallography. I've been reliably informed that knowing the structure of a protein is a very important thing, and that we want to do that. Well, not me, but other people want to do that. Now, myoglobin, like all proteins, are polymers of amino acids. So myoglobin itself has 153 amino acids linked together. It forms the sequence, and then other things happen that make determining the structure difficult. Anyway, we won't talk about that. I don't know anything about that either. So it's also very large compared to what I'm used to. So I study astronomical molecules, which are not astronomical in size, but rather location. So typically a few atoms. So this molecule, this protein molecule, is very large. Now, what about these amino acids? There are 22 proteinogenic amino acids. So what do I mean by that? Well, the ones that are used by life to make proteins. Where does the number 22 come from? Well, there are 20 encoding proteins and 2 special ones, apparently, that can get incorporated into proteins. So 22. And these amino acids, most of them, are chiral. What does that mean? Chiral. Well, a chiral molecule, things that are chiral are non-overlapping mirror images of one another. So like our hands are non-overlapping mirror images. So we can talk about these molecules having a left or a right-handed form. Now, this is important in the context of amino acids because life uses almost exclusively the L enantiomer, the left-handed form, you could say, of the molecule. Okay. There are a few exceptions, but as a general rule, you can just say that life only uses the L enantiomer, effectively. And these molecules, these amino acids, are linked by peptide bonds, CN bonds. It's a little bit more technical, but anyway, you get the idea. This is glycine here. Anyway, this is an astronomer's laser pointer. This is glycine, and there's proline. So glycine is the simplest amino acid. Proline, slightly more complex. You see this heterocycle right here. Okay, amino acids. Let's go on to DNA and RNA, which, as you can see here, is somewhat more complicated. So here you see ribosomal RNA in brown, and for comparison, for size comparison, in blue are proteins, which are sort of dotting the surface of this. So DNA and RNA, what are they made of? Not amino acids, but nucleotides. So here's a picture of a nucleotide as you might find it in a biochemistry textbook. There are 3 main sections to a nucleotide. There's a nitrogenous base or nucleobase, there's a sugar here, and then there's a phosphate group here. Okay, so let's talk about these particular nucleobases. Okay, they form the language of DNA and RNA, the language of genetics. So DNA and RNA use the same, except for this last one here. RNA uses uracil, and DNA uses thymine. All right, so this is the end of the biology. Okay, so probably made a number of mistakes, but what I'm going to be talking about instead is not biology per se, but rather prebiotic molecules. So what do I mean by prebiotic molecules? Well, this is a somewhat nebulous term. Nebulous not in that they necessarily occur in nebulae, but which I would argue, well, I don't want to spoil it. So prebiotic molecules. So what do we mean? Like amino acids, nucleobases, nucleosides, and a nucleoside is basically a nucleotide minus the phosphate group here. But we would say that those are prebiotic. They're not biological molecules, but they're on the road there. They could, under certain conditions, become biological molecules. As well as something like formamide. And why is formamide here? And what do these arrows mean? Well, it means that formamide, there are chemical pathways under certain conditions whereby you can go from formamide to each of these. So we would say that formamide is a very likely precursor for at least some of these, and we would lump it into this prebiotic molecule family. Okay, prebiotic molecules. So how did they form? So how do you get the prebiotic molecules, which are presumably a necessary condition for forming biological molecules? Well, here's the terrestrial view. We've talked about experiments that have tried to recreate the conditions under which life formed. Well, the most famous of them would be the MILLER-UREY experiment. You're probably all familiar with that. So Miller, working under the supervision of Urey, decided to try to reproduce the conditions of early Earth. So they tried to reproduce the atmosphere with a little ocean and then to shoot sparks through it to try to correspond to lightning. So what did they find? Well, what did he get? What happened here? Well, you see here. So what's going on in there? Well, no cells, no life, not even any proteins or DNA or RNA. So nothing crawled out of the flask after the experiment. Didn't have to have any interventions in the laboratory. So what did he get? Well, he got the building blocks. He got amino acids. So amino acids, the building blocks. How many? Well, between maybe 10 to 40. People are actually still analyzing the results of the experiment, but somewhere in that range. Now remember, I've talked about the handedness of amino acids. The handedness of amino acids. So most chemical reactions will give you an equal amount of the left and the right-handed form. You get what's called a racemic mixture. It's very difficult to not get a racemic mixture, and people spend a lot of time trying to figure out how you don't get a racemic mixture of them. Well, what did they get here? What did the Miller-Urey experiment get? Well, they got a racemic mixture, so they got equal amounts. So this does not really inform how maybe life uses only exclusively, or almost exclusively, the L enantiomer. So, and there's another problem with the experiment, which is that it's no longer thought to be representative of the gas composition of early Earth's atmosphere. So we've learned more, we have different ideas. We think there's less hydrogen, basically, than they assumed in this experiment. So there have been others, and I'll talk at the end of my presentation about where research has gone since then. I'll be referencing a lot of the previous talks, so again, you'll hear a lot of evolution. Everyone basically talked about everything I'm going to talk about, but better. So how might they form? What's another view? So we talked about the forming in the early Earth view, which is a possibility. But there's another view, another rather radical view. And what is it? Well, from the title of my talk, you could probably anticipate it. So it's prebiotic molecules from outer space. They came from outer space, maybe. There's a question mark there. Okay. So I'm going to try to whip you up into a state of frenzied excitement over this possibility by showing you something which is perhaps the most riveting picture that I could come up with. So I hope that you're fully fortified from lunch for the shock. So here you go. All right, so it's a very exciting picture, and so I hope you all are enthused. Great, okay, my talk is complete. Okay, well, what kind of rock is this? Well, as Simon Conway Morris pointed out, this is an interesting one. It's a Murchison meteorite. Okay, now we're in Washington, D.C., famous for its museums. If you walk down to the National Mall, you can go in one of the Smithsonian museums, and you can see a chunk of the Murchison meteorite. You can see it, it looks like that. This is at the Museum of Natural History there. Okay. So what is interesting about this rock? Well, it's a rock from space. Well, that's interesting. But another thing that's interesting about it is that from this rock, one can extract some interesting things. You can extract amino acids from this rock. You can extract from a rock from space the building blocks of proteins. Probably did not think that a rock from space would contain something like that. There's some subtlety there. You can ask me about it. I'm going to gloss over it. But anyway, amino acids were extracted, to use the scientific language. Okay. Which ones? Well, these. So notice that we get the simplest one, glycine, but then also some of these others, including this one with the ring here. There's non-trivially complex building blocks of protein in a rock from space. So that's in itself rather interesting. Okay, let's go back to the handedness problem. Remember I told you that it's very hard not to get an equal amount of left and right-handed amino acids or any chiral molecule in a chemical reaction. And yet life, for some reason, decided to go with the L enantiomer. Well, do you have a racemic mixture of these? And the answer, very surprisingly, is no. You don't. You have an excess of the L enantiomer, the one that's used by life. So what can we say about that? Well, you can use the language that you use in papers. You can say suggestive. It's very suggestive. Okay. So this is a very suggestive finding. Okay. Well, now we're talking about amino acids and proteins, but as has been mentioned, the dominant view of the origin of life nowadays is the RNA world view, that you get RNA first, not proteins. Because RNA can self-replicate. Proteins can't self-replicate, as far as I know. We're eager to be corrected, but anyway. So they think the RNA comes first. So what do you need for RNA? Well, we've talked about all of the basic units of an RNA molecule, and one of those is nucleobases. So how can our space rock help us? Well, you can maybe anticipate this. Well, they found nucleobases, or you can extract nucleobases from the Murchison meteorite, very interestingly. So which ones, you may be asking yourselves? Well, these. Okay, you can extract these from the Murchison meteorite. So from the Murchison meteorite, a rock from space, you can extract the building blocks of proteins and a major component of DNA and RNA. Very interesting. So these are meteorites here. This is a meteorite, carbonaceous chondrite. What about comets? This is a comet. This is comet 67P CG, visited by the Rosetta mission. There was, on the Rosetta probe, Rosina mass spectrometer, which analyzed the material coming off of the comet. And what did they find? Well, again, very suggestively, they found an amino acid and other prebiotic molecules coming off of this comet in the middle of interplanetary space. What did they find? Well, they found glycine and methylamine and ethylamine coming off of the comet. So one of the building blocks of proteins streaming off of a comet in interplanetary space. It's very interesting, I think. So now you're probably wondering, how do you get these molecules in space? Well, the answer, I think, is suggested by laboratory experiments. What do I mean? Well, how are you going to make amino acids in space? Well, let's say you take ammonia and acetic acid and cool it down to 25 Kelvin and you irradiate it. What do you get? Well, with electrons in this case. Well, you get amino acids, for instance. You get glycine. You get other interesting things. There are many experiments of this kind, and they show that you can get prebiotic molecules doing this. Well, what does this have to do with space? Well, ammonia, acetic acid, these are some components of cosmic ice, as you might find it on, well, as I'll talk about it, cosmic dust, interstellar dust, but even, for instance, on the ices of outer solar system bodies. Okay, and it's not just water, predominantly water, but not just water, also carbon monoxide, methanol, methane, formaldehyde, so it's very heterogeneous. So all this corresponds to ice. So there's an interesting equation, so to speak. There's ice plus radiation equals prebiotic molecules. Seems to be what's suggested by now a large body of experimental work. Okay, so let's go from the lab to space. So how does this translate? How does the laboratory work translate to space? And how might these conditions obtain? Well, let's talk about stardust. So again, I cannot promise you anything artistic. This is supposed to be stardust, but it could also be a potato or a truffle if you're fancy. So it's a dust grain, a stardust grain. Okay, poetic. So the stardust is in a cloud, in a molecular cloud that has not just dust but also gas. So it's maybe one of the more interesting talks about dust and gas that you hear this week. And so the dust grains interact with the surrounding gas. How do they do that? Well, you have molecules coming in from the surrounding gas, colliding with the dust, sticking to it, accreting to it, as we would say. All right, well, let's say we've got two things on the dust grain. We're going to show you, not going to show you any equations. I'm going to show you a very simple cartoon showing how the surface of the stardust can actually facilitate chemical reactions. It's very sophisticated, so watch this. So the things move around on the surface of the dust grain, and then if they interact, if they encounter one another, they can react and form something new. So that's a surface chemical reaction. A little bit more complicated, but you get the idea. So what happens over astronomical timescales? Well, you have things like this happening. You have more accretion from the gas. And what does that lead to? Well, it leads to a layer of stuff around the dust grain. It leads to, in fact, a molecular ice. So these stardust grains and molecular clouds become frosted. Okay. So we've got the ice. Remember that simple equation before. Where's the radiation? Well, radiation exists in these clouds in the form, significantly, of cosmic rays. I'm going to talk about cosmic rays, but also in molecular clouds, cosmic rays also drive the formation of UV photons, the ultraviolet light. So you've got cosmic rays and ultraviolet light. Now I'm going to have a brief digression on the history of cosmic rays and what they are, just a little bit, because it's an interesting topic, and I think that you all will find it so. Okay. They were discovered accidentally by Father Theodor Wolff, the Jesuit, in 1910. So he was interested in a theory that was going around that people like Becquerel and the Curies thought, which is that radioactivity was just a property of terrestrial material. So he thought, well, if you take measurements at the ground and then somewhere that's above the ground, you should find that the amount of radioactivity decreases if it's just a property of terrestrial material. So he tests that. He takes this Wolff electroscope, makes some measurements at the ground, and then he decides to go to the tallest structure in the world, which is the Eiffel Tower. It was the Eiffel Tower at the time. So, difficult scientific assignment. Goes to Paris, climbs to the top of the Eiffel Tower, takes the elevator probably, and makes some measurements there. So what does he find? Well, in fact, the amount of radioactivity does not decrease. If anything, it may have increased a little bit. So he says, well, maybe there's some radiation coming from space. And like many groundbreaking discoveries, this was thought to be an impossible conclusion. And so it was ignored for the most part, except by people like Victor Hess. He decides to test this by going even higher, taking a Wolf electroscope, making measurements even higher than the Eiffel Tower. So what does he do? He goes up in a balloon, goes up to 5 kilometers, and he takes data. These are his data here on the left. He shows that, in fact, the amount of radiation is increasing very markedly as a function of altitude, showing that absolutely there is radiation coming from space. And then there was follow-up work by Kohlhörster, who showed that indeed this just continues. Radiation is coming from space. Now there is a very nice number of Benedictines here, so I'll say that Victor Hess came to the U.S., taught at Fordham. One of his students was Stanley Yaki, Father Stanley Yaki. Some of you may have even met Father Stanley Yaki, in which case I'd very much like to hear your experiences. But anyway, so student of Victor Hess. Okay. Cosmic rays, where does the name come from? Well, it comes from Robert Millikan. Robert Millikan, pictured here next to Einstein and Father Georges Lemaitre, thought that cosmic rays were photons, so he called them rays. Okay, cosmic rays. There's only one problem with this, which is that, in fact, it's incorrect. They're not photons, they're charged particles. Bruno Rossi, who's at our Cetri Observatory in Florence, did some experiments that showed that they were perturbed by the Earth's magnetic field. So he had this interesting finding. Now, there was a very nice paper that generalized his results and presented the first overall picture of cosmic rays, which was by, believe it or not, Father Georges Lemaitre. This paper, Lemaitre and Vallarta, 1933. Okay, so an interesting connection there. So that's a tangent. So we started talking about cosmic rays in the form of dust, and to dust we are returning in our discussion. So we're going back to cosmic rays. Any rotten fruit that's still in the back, you can send it this way. Okay. So we're returning to our discussion of dust. So how do cosmic rays potentially drive the formation of these prebiotic molecules? Well, they do interesting things. So here's my artistic representation of a cosmic ray shooting through a dust grain ice mantle and forming what's called a track, a radiation track. So this is my representation. So along this track, what's happening? But the molecules are being broken apart. Chemistry is happening. So here's a slightly more detailed view of the track. Okay, so this is what the radiation track looks like in condensed materials, water, solids, etc. Okay, and it causes the formation, again, of reactive molecules, or excited species, things like that. Now somehow, and I'm going to elide over a significant amount of current work and no little controversy in the field, these reactive species will probably end up forming in dust grain ice mantles prebiotic molecules. So that's the connection between cosmic radiation and interstellar prebiotic molecules. Okay, so how do you get them from interstellar space to developing planets? Well, it's through this image. There's Karin in her very nice talk showed a more artistic view of this. So this is the regions that we're interested in are these dense clouds here. So this is where what I've been telling you about is probably occurring. And these dense clouds, as Karin mentioned, begin to collapse and form star and planet forming regions. And so the material formed here on dust grains could get introduced to developing worlds, Earth, through, for instance, comets. All right, how is that? How do you go from dust to comets? Well, what is a comet? A comet is a dirty snowball. So what is the dirty snowball made of? Well, substantially, we think these icy dust grains from the primordial material from which the solar system formed. So we think that comets are, in a sense, molecular time capsules that give us an unparalleled view into the material from which these stars and planets form. So they're very interesting. Okay. Material formed on the dust grains could then get stored in the comets and delivered to, as Darwin called them, warm little ponds. So we still use this term in astrobiology. WLPs, it even has an acronym, WLPs, warm little ponds. And so they're thought to be favorable because if they dry out and get wet again, then this can aid somewhat in the polymerization of these building blocks. Okay, so Darwin talked about that. However, so let's just take a step back. So we can make the building blocks of life. So where does that get us? Can we put them together again? The answer is, so far, in the negative. We cannot put them together again. We try as hard as we can in the laboratory. We try to recreate different sets of conditions, and so far, we've had not any significant results. We can make chains of various lengths of these, but even not necessarily approaching what you would need to have a basic protein or RNA molecule. So it's very difficult. So we've had no success doing that. Although there has been a lot of research into specific things, like how are these molecules UV stable, or what are the role of clays or various other substrates potentially that could help them link together. There's been a number of research on that. But so far, what's the state of the art? Well, this is from a review article in Nature by John Sutherland, who is a very famous person in the origin of life chemistry. And here's his picture. So this is Luca. So it's not a person, it's the last universal common ancestor. This is the sort of fully alive line that he puts here. So where is the state of the art in 2017, which is, we're not, well, basically in the same place. Well, he says we're right here. We're right there. So clearly, we're a long way from doing anything like real synthetic biology, making cells. We're in this fairly primitive state. Now, we may find some conditions that can help us to move beyond that, but as of right now, this is where we are, which tells us that forming life, it's more difficult than we might have thought before. So on that rather optimistic note, I will end my talk. So thank you very much for listening. I'll be happy to take any questions you may have. Yeah.
Okay, we have 5 minutes for questions. Maybe I can encourage also more questions from students too.
Hi there. I'm really curious about the enantiomeric excess of the amino acids on the meteorite, and I guess if you have any explanation for why that might be.
Right, this is a good question. I wish I had a good answer for it. We really don't know, although some people speculate circularly polarized light from some astronomical objects could preferentially destroy the R enantiomer or the D enantiomer, and so just leave the predominance of the L. So that's the best explanation that I know of so far, but it is, I think, very much an open question because it's unusual for that to happen. So.
Hi, maybe this is an of course kind of question, I'm not sure, but you mentioned finding these abundances of prebiotic molecules in the meteorites. Are these same abundances are also found in just terrestrial rocks? What do we find on Earth?
No, I don't think you'd find this in terrestrial rocks. There may be, I think we have some geologists and they can correct me, but I think that these meteorites, these carbonaceous chondrites are unique, though I'm happy to have anyone correct me or preferentially affirm my answer.
Hey, thanks for your talk. There's a lot of steps between methane and a nucleoside or base, and is there any work on going from, I mean, we had the MILLER-UREY experiment, which I loved when I first heard about it, and now I'm like, okay, but that would imply that the bases and these amino acids are sort of an energy sink, that's where the molecules tend to go. And if I told my organic chemistry students, yeah, just put some, methane and some ammonia in a pot and you'll get DNA. Well, first of all, I'd have to fail them on that test, but, is there any work done on going from these small building blocks to these more complicated molecules? Are people looking at, because it can't be a biochemical pathway on the stone, on the rock.
Right. It's not, yeah,
So-
very good question, yeah. So the answer is the chemistry is complicated. So it's not that per se that these molecules are probably sinks, but that under these conditions, let's say you irradiate this low temperature volatile material like ammonia, acetic acid, you'll get some steady state abundance where the radiation will end up destroying, initially forming them, and then you'll get some steady state abundance. Now, what are the pathways that lead to that? We have some idea, but there are a lot of uncertainties, and in fact, we're trying to flesh out, how can you go from simple precursors to these more complex species? And we have some initial chemical networks, basically, but definitely something that needs a lot of work. So.
What are the relative concentrations of the compounds found? Is it a minuscule amount of these prebiotic materials versus a large amount of other carbonaceous or,
Yeah, so I'm not so sure about in the meteorites themselves, but on stardust grains it would maybe be a millionth the abundance of molecular hydrogen or 10 millionth the abundance of molecular hydrogen. So we're talking about trace amounts, but in the context of space, if you accumulated a lot of it altogether, you might have a significant amount that could make its way in early Earth or early planet conditions to some kind of warm little pond. But again, whatever those conditions are, we've yet to find them. So.
Okay, I'm going to do one more question.
Paul Giesting, Wyoming Catholic College. Actually, in response to the comment about, I don't know if I'm the only geologist in the room, but I'm one of the few, I guess. One of the reasons why your work is so important, why actually we have to look to places like outer space to find the origins of life, is that the surface of terrestrial planets is an oddly hostile place for biomolecules, prebiotic molecules to form because they're very reduced. I mean, the Miller-Urey experiment, as I recall, was really kind of like, well, there's Jupiter and Saturn. Let's put Jupiter and Saturn in a pot and strike it with lightning and see if it works. Yes. But Jupiter and Saturn are bad examples. Terrestrial planets, even when they form, are somewhat oxidized. And then, of course, we've had this great sort of, life has sort of pulled the ladder up after it because life has now filled this atmosphere with 20% oxygen, which is an insane, chemically insane state of affairs that only life could produce. Only life is powerful enough to do weird things like that, so that those type of molecules, we find these type of prebiotic molecules in geologic and say shales or something, gosh, we almost take it for granted that life put them there, because of course life is so ubiquitous on the current planet that we do find those molecules in rocks like that, in low temperature rocks that haven't been cooked to very high temperatures, but we're almost certain that life put those molecules there. So we have to go back to astrochemistry to give us these sort of answers. So it was a great talk. Thank you.
Oh, well, thank you. Yes.
All right, maybe we can give our speaker another round of applause.